Hey everyone, welcome to our first lecture for MLT205 blood banking. This course will be taught virtually and all lectures will be recorded via Zoom and uploaded so you can listen to them as many times as you would like. For our first topic, we will be covering Chapter 2 in our textbook, Blood Banking and Transfusion Practices by Paula R. Howard. And so we'll get right to it. So the topic today will be an introduction to antibodies, the immune response, and complement. So there are some objectives that go along with the PowerPoints. By the time you're finished with the lesson, you will be able to define terms in relation to red blood cells, which throughout the course will be abbreviated as RBCs, and transfusion. And so these topics include antigens, immunogens, epitopes, and antigenic determinants. You should be able to describe the characteristics of antigens that are located on red cells, white cells, and platelets. Diagram the basic structure of an immunoglobulin class G or IgG molecule and label important parts, including heavy and light chains, FAB, and FC regions, variable region, hinge region, antigen binding site, and macrophage binding site. For our second page of objectives, you should be able to compare and contrast immunoglobulin M, also known as IgM, and IgG antibodies. How are they different in structure, function, and the detection temperature for agglutination reactions for each? Distinguish the primary and secondary immune responses with regard to immunoglobulin class, the immune cells that are involved, level of response, response time, and antibody affinity. And accurately grade and interpret agglutination reactions using an agglutination grading scale for antigen-antibody reactions performed using a tube method. At the end of this unit, we will also get into complement proteins, and so this last page of objectives has to deal with them. So when you're finished, you should be able to compare and contrast the classical and alternative pathways of complement activation, review the steps of the classical pathway relevant to blood banking, and outline the biologic effects mediated by complement proteins in the clearance of red blood cells. So those are all of the objectives for this unit. And everyone should also keep in mind that it is important that you do pay attention to these PowerPoints and make sure you understand the concepts, because part of this class will also involve you taking a short quiz on the information included in the PowerPoint at the end of the unit. So for an introduction, when you hear people refer to immunohematology, this is simply another term that's used to describe the blood bank lab, okay? And so if you break down immunohematology, you probably remember me saying this to you guys before. Whenever you see that word immuno, that prefix, it is related to antibodies. Okay, so that is another way that blood bank is described, as the immunohematology lab. In the blood bank, we study blood group antigens, which will be abbreviated as capital AGS from now until the end of the class, and antibodies or immunoglobulins, which will be abbreviated as capital A little bs, and their importance to transfusion of blood components. So there will be a focus on red blood cell antigens, and red blood cell antigens are simply markers that are embedded in the cell membrane of the red cell and their corresponding antibodies. And there are over 30 different red blood cell antigen systems. And we will be focusing on the most important ones of those systems as we get through our course. So let's talk a little bit more about antigens. So an antigen can be defined as a molecule that binds to an antibody or T cell receptor. So maybe you recall from hematology, MLT155, hopefully you do. T cells are a type of lymphocyte that are responsible for tissue immunity. And anybody remember where they are formed? Remember our rule to remember? Hopefully you do. T means they're developed in the thymus. Okay, so they don't develop in the bone marrow like our other blood cells do. And so T cells are very important in presenting antigens, okay, to other cells so that antibodies can be formed. There are two other terms that are important on this slide. Well, there's more than two, but the next two. Whenever you see allogenic, allogenic describes non-self antigens. Okay, so these can be from bacteria, viruses, or red cell markers from another person. Okay, so we will be focusing on, red blood cell markers from another person in this course. And so when an individual receives a transfusion of red blood cells from a donor, they're technically getting red blood cells from another person. Okay, and so we will call that allogenic. When a woman is pregnant and she's carrying a fetus, the fetus also can have antigens that are somewhat different than the mother's, because remember, the father's information also goes into contributing to the antigens that that fetus would have on its red cells. So those are the two types of allogenic situations, transfusion or a woman who's pregnant with a baby. When we compare allogenic to autologous, that prefix auto means self. So these are self antigens. Okay, auto means self. So a couple of other terms on this slide, and then we'll move on. When you hear the term hapten, this is a partial antigen that actually requires a carrier molecule to elicit an immune response. Okay, so what the heck does this mean? So eliciting an immune response, let's look a little bit more carefully at this first. When you are exposed, or a person is exposed to the red blood cells from another person, either through transfusion or a woman who is pregnant with a baby, there is a chance that that person can develop antibodies against antigens that are present on the red cells of another. Okay, and so basically that's what we call eliciting an immune response. Okay, so in essence, what can happen, let's take the example of a person who's receiving a transfusion of red cells. And we'll be first dealing with the ABO-RH system, which many of you, I'm sure, learned a lot about in biology, or may recall us talking about in phlebotomy a bit. But beyond ABO and RH, there are other antigens that are present on red blood cells. And so each time a person is transfused with the red cells from another, even if it's the same exact blood type, ABO-RH blood type that that person has, they're still exposed to other antigens present on the surface of those red cells. And there is a chance that when the body sees these red cells from a donor, they recognize them as non-self. And they say, hey, that's not my antigen, I don't have that. And the person is basically stimulated to make an antibody directed against that antigen. So that's what we talk about eliciting an immune response. So an immune response is when an individual makes an antibody against an antigen. Okay, so a hapten actually requires a carrier molecule such as a medication in order for that to happen. So it's a partial antigen, it's not completely whole. Another important thing about antigens is that they contain different antigenic determinants, and these are called epitopes. And each epitope can elicit a different type of antibody. Okay, so think of the antigen as having multiple pieces to it. Okay, and so the piece that is recognized as foreign, okay, an antibody may be made by a person directed against that piece or epitope. Okay, but there's definitely more than one. All right. Okay, so in transfusion medicine, most reactions are humoral, and that is, hopefully you remember, when we say humoral, we're talking about B lymphocytes. So you may recall the B lymphocytes. If we were meeting face to face, I would ask you guys this question: where did they develop? Hopefully you know. I think B is for bone marrow. So the B lymphocytes develop in the bone marrow with our other cells. You may recall from last semester that plasma cells are B cells which transform into the plasma cell, basically to make antibodies. So plasma cells are antibody producing cells. Some B cells will move on to become memory cells. And memory cells are basically B cells that hold on to how to make a specific antibody, so that when a person is exposed, let's say to a foreign agent, an infectious agent, a second or even a third time, the memory cells are able to pump out that antibody much more quickly because they remember how to do so. So here we see next a little diagram from our book, and this is just going through the process a little bit about what happens to trigger antibody production. So, we start off here with a B lymphocyte that's recognizing an antigen, something that's non-self in this case, okay? B lymphocytes present the antigen to the T lymphocytes, and then the T cells will trigger the B cells to produce plasma cells. And remember, the plasma cells actually make the antibody, and then some of those B cells will become memory cells, so that on subsequent exposures, if that happens down the road, the antibody can be produced much more quickly, and the T cells don't even have to be involved anymore, okay, because these memory cells remember what to do. Next, we see table 2.1 from our textbook, and these are factors that contribute to the immunogenicity, okay, of an antigen. And so you might say, well, what the heck does immunogenicity mean? And so it's up here on the top of the slide. Immunogenicity basically refers to the ability of an antigen to evoke an immune response. And remember, evoking an immune response is when we are making that specific antibody that would be directed against that antigen, okay? So what factors contribute? Let's look at a few. First of all, the chemical composition and complexity of the antigen. So antigens that are proteins are best at stimulating the immune response, okay, followed by complex carbohydrates. Next, the degree of foreignness. When that immunogen or antigen is recognized as very different from self, there is a greater likelihood that we're going to make an antibody directed against it, because it's basically detected as being non-self. Size of the particle does matter, okay? So molecules or antigens that have a molecular weight greater than 10,000 Daltons are better at eliciting an immune response than smaller molecules. Okay, so think about it. Size, our body is going to recognize it as foreign, and we're going to pump out an antibody against it. A couple of other things: dosage and antigen density. The number of antigens that are introduced, obviously the more we're exposed to, the better likelihood that an antibody will be formed. And then lastly, the route of administration. Okay, and this would be the manner in which the antigen is introduced. Okay, so this, very appropriate to right now, would be related to, for instance, effectiveness of a vaccine. Okay, and so right now in the middle of our COVID-19 outbreak, in the news every single day is, will scientists be able to develop an effective vaccine soon? Okay, and so basically, most vaccines, think about this in your own life, are given through injection. So intramuscular into the muscle. Think about your annual flu vaccine, influenza vaccine. It's in the upper arm, okay, and that's intramuscular. Intravenous would be directly into the veins. But these have been found to be better routes, okay, for the antigen being recognized by our body as something foreign, and for us to elicit an immune response. And just, I can't say it enough, remember, eliciting an immune response means making an antibody against that antigen that's present in the vaccine, okay? Okay, so we're going to move on to a couple of other important things related to the immune system. First of all, active immunity. Active immunity are antibodies that are formed when an individual is stimulated by an antigen, and this could be done in a variety of ways. So you have an infection, some foreign agent entered your body, and your immune system is stimulated to make an antibody against it. Even the last example with the vaccination, okay? Every year when you get your flu vaccine, scientific experts try to make an educated guess each year which strains of the flu are most likely to be the prevalent ones causing infection. And so antigens of those weakened attenuated pieces or antigens of different flu virus go into that vaccine every year. Okay, and so basically, when your body recognizes those foreign pieces of the virus, it makes antibody against it. Okay, and that is what offers us protection. So what you're seeing hopefully in each one of these thus far, active means the person's own body is making the antibodies. Okay, through transfusion. So how would this happen, you might say? So I spoke a few minutes ago of how when a person is given a transfusion of red cells from a donor, even if it's the exact ABO and RH type of that individual, there's still a chance that the recipient of those red cells could become sensitized, okay, meaning recognize certain other antigens present on those donor red cells and make antibodies against them. Okay, so that's active. Okay, and then pregnancy, similar thing. So once again, as I mentioned previously, when a woman is pregnant with the fetus, in addition to her own genetic information, there is information from the father that predicates which antigens are present on the fetal red cells. And so if through the pregnancy there is mixing of the fetal blood into the maternal circulation, the mother may come into contact with an antigen on the fetal red cells that she lacks. And in this case, her body may say, hey, I don't have that antigen. I'm going to make an antibody against it. Okay, so once again, it's active immunity because in each one of these instances, an individual's own immune system is making the antibody in response to exposure to the antigen. When we compare passive immunity, this is when antibodies from another offer protection to the individual. So their own body is not making the antibody in response to the antigen, and so it's a passive situation. So there are two examples that are, that I can give for passive immunity at this point. So getting back to that pregnant woman, all mothers, okay, basically, when they're pregnant, certain class of antibodies, namely immunoglobulin G, has the ability to cross over the placenta from the mother's bloodstream into the baby's bloodstream and offer protection. Okay, so it's only class IgG. And we'll learn about these classes and how they're different in a few minutes. So basically, why is it passive? Because the baby did not make these antibodies on their own. They originated from the mother. So it's called passive immunity. Currently, with everything that's going on with the coronavirus, there have been some interesting cases where convalescent plasma has been transfused into people who are severely ill with COVID-19. And so I want to talk a little bit about convalescent plasma. And you may have heard about some of these stories on the news if you've been watching any national news. We've had some local cases as well where this has worked. And so basically, you know, we don't know why at this point, but some people who are infected with the coronavirus have a milder form of the disease, which doesn't necessarily require hospitalization or use of a ventilator. And there is a significant portion of the population who may have had these mild symptoms or may not even have realized they were infected at all, and they have developed antibodies against COVID-19. So convalescent plasma can be withdrawn, okay, taking the plasma of someone who's recovered from the infection, and then taking that person's plasma and transfusing it into someone who has an active, life-threatening COVID-19 infection. Basically, this would be passive immunity because we're giving that person who's really sick the plasma from another that contains antibodies that that donor made in response to the infection. But there has been some success with the use of the convalescent plasma in helping to cure some people that have had very serious infections. So hopefully it makes sense to you that this would be passive immunity as well, because that person who's very sick in the hospital, you know, is getting this plasma that already has antibodies against the virus in it. So they are not necessarily making their own at this point. It's not to say that they won't make their own down the road, okay, but for now, we're giving them some of the antibodies. So let's look a little bit more carefully at antibodies, and some of you may have learned about this in previous biology courses. So antibodies are basically glycoproteins, and they're composed of 4 polypeptide chains that are joined by disulfide bonds. Okay, and we're going to look at a picture of an antibody in our next slide. Okay, so the basic antibody molecule has 2 heavy chains and 2 light chains. Okay, and the light chains can be either kappa or lambda. So it's either 2 kappa or 2 lambda. There's not a mixing of, let's say, 1 kappa and 1 lambda. It's either all or none, okay, kappa or lambda. Within the antibody molecule itself, there are areas called constant regions, and these correspond to areas of the heavy chains. And this is what determines the class of antibody that we're dealing with. And in the literature, you may hear antibody class referred to also as an isotope. Okay, so there are basically 5. We have IgG, IgA, IgM, IgD, and IgE. Now, the 2 that are really most important to blood bank that we're going to focus on are IgG and IgM. Okay, but we can tell that an antibody is IgG by its heavy chains, and likewise, we could tell that an antibody is IgM by its heavy chains as well. Okay, so the heavy chains defines the class of antibody that we have. Then there are variable regions, and the variable regions occur, they span both the light and heavy chains, and in this variable region, this is the portion of the antibody that is specifically designed to bind a specific antigen, okay? So what the heck does that mean? I'll try to well, when I describe this, it may be something that you've heard in other classes before, but through the ages, antibody and antigen interactions have been referred to as the lock and key. Okay, so what does that mean? It means, hopefully that, my key is only going to unlock my door, okay, and your key is going to unlock your door. And likewise, when we're talking about antibodies, an antibody is specific for one target, okay, and it would not bind so well to a target that it was not designed for. Okay, so here's a picture from our book that hopefully can bring this concept home. And so here we see a picture of a red cell or biconcave disc. And so when you're looking at the surface of this red cell, we see a variety of different antigens or markers that are embedded into the red cell membrane. Okay, and so these are referred to as epitopes, right? Each one is a little different than the other. Okay, and so this rings home what I discussed previously as to how there are antigens can have many different epitopes, right? Okay, so here we see some diagrams of 3 different antibody molecules. And so in blood banking and in science in general, the basic antibody molecule is designated as a Y, okay? And so, we have heavy chains and then disulfide bond and light chains. And so when you look at the end of the antibodies here, these are the binding sites, okay? And this is where it's specifically designed to a certain target antigen, okay? So let's look. Okay, so we see the structure here of this binding site. Well, this antibody is going to fit much better, okay, with this epitope than it would, let's say, with the pyramid here or the triangle here, okay? And so when we look at our second, we can see here how the binding site here is specific for this epitope. And lastly, we have a circular one here, which is going to bind to this epitope. And so hopefully, this makes sense to everybody that, they are specific to a certain antigenic determinant or epitope. So let's look a little bit more carefully once again at the structure of the antibodies. Okay, and so here we see a diagram from our textbook. And so we can see how we have the heavy chains, okay? And the heavy chains are, attached with, these disulfide bonds, and they call that the hinge region, okay? And then on the inside, we have the light chains. And remember, the light chains are going to be either kappa or lambda, okay, chains. And then basically, we looked at the last slide of the FAB region. And FAB region simply means fragment antigen binding, okay? And so that's at the very end, and you can see how both the heavy and the light chain contribute to that FAB, okay? So it's going to be the same on each side. And so when you look at this picture, you could see that a basic immunoglobulin molecule, and this would represent immunoglobulin G or IgG, basically has 2 antigen binding sites, okay? And so that's important to remember. Okay, so IgG has 2, okay? And so that's FAB in the Y region. When we look at this area of the basic antibody molecule, we can see that there's another area here called FC. And so FC, this is very logical. You see that C binds something called complement, okay? And it's also referred to as fragment crystallizable. It's all a bunch of C's. So what you need to remember here, okay, it's not the same as FAB. And basically FC binds complement, which are a series of proteins that we'll get into more detail about towards the end of this unit. Okay, so what do we need to take away here? Our basic antibody molecule has heavy chains, okay, and the heavy chains tell us which class of antibody we have. It has light chains, 2 light chains, which are either kappa or lambda. And the heavy and the light chains go together to make the antigen binding site, which is called FAB, and our basic antibody molecule has 2 of them. Then when we move down to this arm of the molecule, we have our FC region, which binds complement, and this is known as fragment crystallizable. And this is what IgG would look like. So let's move on and talk about the other class of antibody that's relevant to blood banking, and this is class IgM, or immunoglobulin M. Class IgM is what we call a pentamer, or pentamer, however you want to say it. And when you look carefully at this diagram, you see that, okay, here we have, this is a good one to look at, we have one of those basic antibody molecules that we looked at in the last picture, okay? And so IgG, we already said, would be represented by this picture here with the dashes, and it has 2 binding sites. When we look at IgM, we could see that it has 1, 2, 3, 4, 5. Okay, it has 5 basic immunoglobulin units that come together to make the IgM molecule. So 5, if you remember, your prefix is penta, means 5, so it's considered a pentamer. So because it has 5 of these basic units joined together by something called a J chain, okay, you know, we could say, well, if a basic unit has 2 binding sites to it, and this one has 5 immunoglobulin units, then we expect to have 10 total antigen binding sites in IgM. So if you count them, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So that's important to remember. So it's a pentamer, has 5 immunoglobulin molecules coming together to make IgM, and that means it has 10 FAB binding sites. So this is table 2.2 from our textbook, and this is a good comparison of IgM and IgG. I'm going to talk a little bit about some of these things that are most important. So of course we know that the heavy chains tell us which class of antibody we have, and so IgM has mu and IgG has gamma, and hopefully that's logical. You know, we know they have either kappa or lambda. The J chain, remember, is what joins all of those 5 together for IgM, and because it has 5 units coming together, it's going to have a higher molecular weight. Okay, so when we look here, 900,000 compared to 150,000. Okay, valence is how many antigen binding sites. So remember, 5 times 2 is 10. IgG only has 2. Okay. Let's see. IgG is the one that really provides long-lasting immunity. We're going to learn IgM rises and falls very quickly. And then something else that's very, very important is, can this class of antibody cross the placenta from the mother to the baby? And when we're looking at IgM, the answer is no. And here's how I remember it. Think, it's huge. It's a pentamer. It has 5 big, 5 individual molecules going in to make it. So it's very, very large. So I just think it's too big to cross over. However, IgG can, and thankfully it's able to, because that's how we can have passive immunity passed from mother to baby. Okay, and so IgG, yes. We're going to talk about complement a little bit later. Both IgM and IgG can activate complement, but IgM is definitely more efficient at it. Okay, and it's just because it's so huge. Okay, it's very, very big. When we're detecting these classes of antibodies, they react better at different temperatures. So when we're detecting IgM class antibodies, they react best at immediate spin. And immediate spin means that we basically add a serum or plasma source with the antibody, with the red cells, and we centrifuge immediately, and we look for agglutination. That's what we call immediate spin, and this all happens at room temperature. Okay, so IgM, you want to think room temperature. IgG reacts best at 37 degrees, which requires an antiglobulin test, which is a test we'll talk more about later. And 37 degrees C is the same as human body temperature. Okay, so room temp versus human body temp. So let's move on and talk a little bit more about the immune response. So there is a primary response and a secondary response. So we'll start with the primary response first. So primary response is how our body reacts the first time we're exposed to an antigen. There is a little bit of time, anywhere between 5 to 10 days, before our body is able to start producing antibodies in a primary response. And what you always want to remember is that IgM antibodies are the ones that are produced first, okay? And maybe this makes sense if we think about IgM as being a pentamer and having 10 antigen binding sites. Wouldn't we want this one to be produced first so that it can try to bind to as many antigens as possible, okay? So IgM will rise and fall, and then IgG, okay, will arise next. IgG levels are maintained over time. So, you know, when you are vaccinated, for instance, you know, we all maintain, hopefully, IgG class antibodies that are there to protect us, okay, beyond through the years. Less antibody is produced in a primary response than compared to a secondary response. So secondary response is how we react when we are exposed a second or even third time to the same antigen. It is faster, okay, so remember we have memory cells. It's going to happen much more quickly, so within 1 to 3 days we're going to have antibodies being produced. And now it's mostly IgG antibodies, okay, with smaller amounts of IgM. And the antibody level is high and sustained for a longer amount of time. So this is a picture from our book that, you know, that shows the primary antibody response side by side with the secondary antibody response. And so, you know, when we look on the left here, we see first exposure, okay, B cells are exposed, they're activated, plasma cells are making the antibody, and you know, first we have our IgM and then the IgG, and we see that this is going to take 5 to 10 days, okay, until we get antibody being produced. When we look at the second time we're exposed to that very same antigen, we now have our memory cells, okay, that are going to pump out the antibody much more quickly. And so look at the amount of antibody. Okay, so much more antibody is produced. It's mostly IgG. And so we can see how, you know, it's going to happen much more quickly there as well. So let's talk a little bit more about antigen-antibody reactions. So when the antibody finds its target antigen and binds to the epitope, the combination is called an immune complex. Okay, so antigen binding to antibody is an immune complex. The strength of the binding is going to be affected by a couple of things. So first of all, the size of the antigen, the shape of the antigen, and the charge of the antigen. So all of those things go into the strength of that binding. Immune complexes are held together also by non-covalent forces, and this contributes to the avidity. Avidity is the word that describes the strength of the attachment, okay? And so a little bit of chemistry comes into play with these forces, okay? So once again, we have a diagram from our book, and this is a comparison of fit. And we kind of looked at this previously. So when we look at the antibody in the first picture, we see the FAB is rounded, and we see how it's going to bind to its target antigen. And this is a really good fit. So there's going to be high attraction, low repulsion. There's going to be strong avidity, strong strength of that that interaction here, because they're fitting like like a finger in a glove, so to speak. When we look at the second example here, this is an example of poor fit. So when we look at the binding site here, we see it's not exactly specifically designed correctly for this antigen. So the strength of this binding is going to be weaker. There's going to be a higher repulsion rate, so low avidity. Table 2.3 in our book outlines different non-covalent forces that are involved with antibody binding to antigen. And so this is where I say, if you remember your high school chemistry class, or maybe some of you had chemistry in summer 1 recently, this is really chemistry in blood banking. And you guys know I'm not a chemist, so we're just going to go over this real quickly. So there are electrostatic forces, or ionic bonds, okay, and this is when opposite charges attract, okay. Hydrogen bonding, okay, when a hydrogen atom is involved. Hydrophobic bonding are weak bonds as a result of the exclusion of water. And then we have these van der Waals forces, and this is attraction between the electron cloud of one atom and the protons within the nucleus of another atom. Okay, so just be aware of them, that they do have an effect on binding, but this is not chemistry class, so I'm not going to ask you specific details about each one of these. Okay, so let's get into a bit more detail about antigens and antibodies. So we already established, so this should be a bit of a review, that in blood banking, antigens are located on the red blood cell surface. They are part of the cell membrane, okay? They are glycoproteins or glycolipids, and they will agglutinate when combined with their target antibody, okay? So we'll be talking a lot more about this in lab as well, but imagine the red cell has its antigens on its surface. When the antibody finds its target antigen on the surface of that red cell, it's going to link onto it, and visibly we will see agglutination. So that's how we know that that immune complex has formed. Some antigens on the red cell surface elicit a stronger immune response than others, and so one that we'll be talking about pretty soon is called the big D antigen. And you might say, what the heck is big D antigen? And this basically is the antigen that confers Rh positivity. Okay, so if your red cells have the big D antigen, you are Rh positive. If your red cells lack the D antigen, which that's only about 15% of the population, including me, moi, you are Rh negative. So by far, most people have this big D antigen. So what does it mean it's capable of eliciting a strong immune response? What this means is, when you have someone like me who lacks the big D antigen, if I were to be transfused, let's say, with red cells from a donor that had this big D antigen on their red cells, there's a strong likelihood that if I come into contact with that D antigen, that my body is going to make an antibody against it, okay, because it's very capable of eliciting that immune response. So let's look at our antibodies. So antibodies are molecules present in plasma or serum. IgG antibodies are considered clinically significant, and this is because they react at human body temperature, 37 degrees C, okay? IgM antibodies usually react at room temperature or cooler temperatures, and they're not usually considered significant unless they activate complement. So an interesting thing about this, the first antigen system that we will be getting into is the ABO system. And ABO antibodies are very strong, and they activate something called complement that will get to at the end of this unit. But ABO antibodies are considered, well, they can lead to serious consequences if they are activated. And so we'll get into more detail about that later. So blood bank is sort of a unique lab because it's one place or one area of the medical laboratory where we basically test a person's red cells, and then we test their serum or plasma. We test them separately. So you may recall from blood banking, I'm sorry, not from phlebotomy, when you did your phlebotomy rotations, most often clinical sites will not allow students to draw these specimens, and the reason is that patient misidentification errors can lead to death. It's very, very tragic, but in our region we've had a few of these phlebotomy-related patient identification errors, not necessarily from a student, okay, but you know, if a person is given red cells that are not compatible with their own blood type, they can die within a few minutes. So it's very, very serious. So common types of tubes that are tested in the blood bank, I think most labs use the pink EDTA anticoagulated tube. We could also use a regular lavender EDTA tube like we use in hematology for the CBC as well. But what that lab is going to do first is centrifuge, and we're going to get the separation, okay, and we're going to have the red cells on the bottom, okay, and remember, red cells, the antigens are going to be present on the red cells, and then we're going to have either serum or plasma on top, and so we're going to remove this with a pipette and try not to mix it up with the red cells, and we test them separately. So the patient's serum or plasma is where the antibodies will be found. Okay, so blood bank is unique because we always test red cells and serum or plasma separately. And so here you see the buffy coat, which is the white cells and the platelets. So one other thing I do want to mention, it is possible, okay, you see serum, for us to use a tube of blood like a plain red top tube. And so in that case, we would have a clot with the cells on the bottom, and we would separate the serum from that. It's a little bit more challenging. Most people prefer using the anticoagulated samples because when you have a serum sample, you kind of have to mash the clot to get the red cells loose from the clot. And obviously, we would not be able to use an SST tube with the gel in it, because that gel would prevent us from getting, you know, to the red cells in the clot in the bottom. Okay, so most labs do use the pink EDTA anticoagulated tube. So let's talk about in vivo reactions. Okay, in vivo means in the human, in the person. And I kind of spoke to this earlier in this unit. So when a person is exposed to foreign antigens on a red cell of someone else, either through transfusion of donor red cells or a woman being exposed to fetal red cells, this can cause something called sensitization. And basically that's the body recognizing that there are markers on these red cells that are foreign and not self, okay? And so this can cause the formation of ALO antibodies. And so ALO antibodies are antibodies directed against non-self antigens. So in this case, the red cells of a donor or the red cells of a fetus, okay? There is a test in blood bank that we do, and we will be doing it virtually later on, called the antibody screen. And so maybe when you did your phlebotomy rotation, you recall people talking about type and screen. So type is ABO and RH, and screen is finding out if this person has ALO antibodies. Okay, so when would a person possibly have these ALO antibodies? Okay, think about it. They were previously transfused at least one time, or a woman who was pregnant at least one time. And the pregnancy does not necessarily need to lead to a live birth. Okay, so if a woman has a miscarriage, she could still be potentially exposed to those fetal antigens. Detecting when someone has these ALO antibodies is really important before this individual gets another transfusion, okay, to prevent formation of immune complexes in vivo. Okay, and so I want to speak to this just for a moment. So if somebody has some ALO antibodies from previous transfusion, or a woman who's had a pregnancy, let's say, we want to make sure we find out the identity of that antibody. It's really very important, because here's why. Let's say they're getting another transfusion. Okay, let's say we transfuse them with red cells that happens to have the antigens to the ALO antibody they have. This would be very bad news, okay, because basically what's going to happen is their ALO antibody is going to find the target antigen on these donor cells, and it's going to lead to destruction of the donor cells. So basically, the transfusion, would not be doing what it's meant to do. And, most often when people are getting transfusions of packed red cells, it's because they've lost blood or they're anemic and they really need those red cells to help carry oxygen. So we want to prevent them from being destroyed, okay? Antibodies can also activate complement proteins, and this can lead to red cell destruction, and we'll be looking at that at the end of the unit. Okay, so let's talk about agglutination. So when we have an antigen and an antibody come together to form an immune complex, reactions that we look at in the lab are called in vitro reactions, or in the tube. And the reason we know the antigen and antibody have come together is by visibly seeing agglutination or clumping of the red cells. So if antigen and antibody did not come together to form a complex, that's a negative reaction, we're not going to see any agglutination. There are two steps that need to happen for us to visibly see agglutination. Okay, so the first one is called sensitization. At this stage, the antibody binds to the antigen. It says, hey, there's my target, and it binds to it. But the fact that this has occurred is not visible to our eye at this stage, okay? But it doesn't mean it didn't happen just because we can't see it, okay? So antigen and antibody have come together, but we are not able to visualize it, which brings us to our next step, lattice formation. So lattice formation, okay, we have our immune complex, in this case, our red cells that have the antibody attached to them. When there is cross-linking of antibody binding sites to other red cells, that's when we start to see visible agglutination. Okay, and so we're going to look at a picture of this momentarily. So before we do, let's talk a little bit about Table 2.5, different factors that affect agglutination. And so when we're talking about the first stage, sensitization, temperature, remember, commit it to memory, very, very important. IgG antibodies react at 37, IgM at room temperature or cooler. Incubation time. Some antibodies are immediate spin, some might need some incubation time. pH, we want absolutely neutral, that's ideal. And ionic strength can affect this as well. When we move on to lattice formation, the ability of that cross-linking to occur, think about a lattice fence, how different pieces of the fence cross over to form the barrier, so to speak. There are a couple of things that are important. We're going to look at each one of these in a moment. Zeta potential is the normal space that exists between red cells. We'll look at a picture of that. Zone of equivalence. Ideally, we want to have the same amount of antigen as we have antibody, and that's where we're going to get ideal formation of immune complexes. And then centrifugation. Okay, the time and the speed that we centrifuge is very important, but centrifugation is a way to bring the red cells closer together to collide so that we can get lattice formation. And this will hopefully be apparent when we look at the next picture. So this is another image from our book, and I really like it. So on the left-hand side, we see our red cells here, and you notice how they all have this negative charge to them. And so even I remember this from chemistry, guys, right? Like repels. Okay, so these guys are going to be like moving away from each other normally. Okay, the negatives are going to repel each other. So when we are performing our testing in the lab and we centrifuge, right, and we have antibodies binding, the centrifugation kind of forces the cells to come together. And look at what we see here. This is really important to lattice formation. We see, if we were face to face, I would ask you guys, hey, what molecule is this one? It looks like a pentamer. Hopefully you're going to say IgM. We could see how binding an FAB is binding to an epitope on this red cell, and then FAB of that same IgM is binding to an epitope on another red cell, and that's what we mean by cross-linking. So that's what's going to enable us to see the agglutination of the red cells coming together. They look clumpy. All right, so you're going to see this more than one time in this class. This is another diagram from our textbook that's really very important. But the ability for the lattice formation is really affected by, hopefully you know these signs, concentration of antibody and concentration of antigen. So as I mentioned previously, ideal is in the middle here, zone of equivalence. And so when we have the same amount of antibody as we have antigen, we're going to get good binding and cross-linking. Okay, so here you can see the red cells, right? And we could see how these are, you know, little diagrams of IgG molecules, right? But we could see FAB is binding to one red cell here and a different one here. And we're getting a lot of good cross-linking there. So we should be able to visualize that agglutination easily. However, when we have either too much antibody or too much antigen, immune complexes may have formed, but we may not be able to visualize them. Okay, so that's the thing. Remember, this is affecting the second stage, lattice formation. So let's look at zone of antibody excess. So when we look at this diagram, we see there is way more antibody present than antigen on our red cells. And so we see that the antibody binding sites are binding to these red cells, but we're not seeing that cross-linking that we see here. And so even though sensitization has occurred, when we look at our test system, it may look negative. We're not able to see it because we have way too much antibody here. Okay, so we're not getting that nice cross-linking. On the other hand, antigen excess, way too many red cells here and similar kind of things. So all of the binding sites are taken up of the antibody that we have, but we're not seeing cross-linking. And so once again, it may look falsely negative. So immune complexes have formed, but if we don't get the good lattice formation, we're not going to be able to detect them. Okay, so this is a nice picture of the lattice formation once again. So we see how the binding sites of different molecules are reaching out and binding to different red cells, and we're going to see that nice agglutination in our tube. So when I finish talking about agglutination, excuse me, and about how reactions are actually graded. The strength of reactions are very important in blood banking. And so when we do our virtual lab, we'll be talking more about this and looking at pictures. And at some point, when we meet face to face again, we will have one or two blood bank labs, just, you know, to make sure everybody understands this. But when we combine red cells, which is our antigen source, in a tube, and the target antibody is added, okay, we centrifuge. Okay, we centrifuge for 15 seconds at 3,500 RPMs. And when we take the tube out of the centrifuge and we hold it and we look at it under a lamp, and we just gently move that tube back and forth, if we see one big clump, one big piece of the red cell button come off the bottom of the tube, this is the strongest reaction we can have. So in blood banking, when we're recording reactions, we don't just say positive or negative. When we have a positive, we need to say, is it 4+? That's the strongest reaction we can have. And so all of the red cells in that tube are in a button, and so the background liquid is clear. Okay, there are no red cells in that background, so that's a 4+. A 3+ reaction might be when we have several large pieces of red cells, but it's not one big chunk. Okay, and here the liquid in the tube is still clear. Okay, because the red cells are bound up into a couple of big pieces. 2+, we're moving into many medium-sized agglutinates, but the background is still clear. 1+, we can have a variety of sizes of red cell agglutinates. However, the background looks cloudy because there are still some red cells that are kind of flowing in the liquid. Okay, so that would be 1+. And then a negative, which is designated by a 0 in blood bank, it just looks like the blood, the diluted blood. Okay, so there's no pieces of anything in there. It's a smooth suspension, and that would be a negative reaction. So these are very important. Strength of reaction can tell us a lot about the antibody that we're dealing with. So that's important. So we're going to go through some tips about routine testing, and we'll be talking about some of these things in lab as well. So we learned on the last slide that agglutination is the main way that we tell whether an immune complex has formed, when we see those red cells agglutinating. However, in blood bank, hemolysis also can indicate that an antigen and antibody have come together to form an immune complex. And so you guys remember from hematology, hemolysis is the destruction of red cells. Okay? So when can hemolysis occur? If something called complement is activated. And trust me, we're going to get to complement in a few minutes. Okay? So hemolysis is normally caused by complement activation. Basically, you know, the fact that the immune complex formed and complement was activated is what causes the red cells to be hemolyzed. So when we're looking at a tube, for instance, the red cell button is smaller and the background liquid is going to look pink to red because remember, some of the red cells were destroyed and hemoglobin was released after centrifugation. Okay? So in blood bank, it's essential that only fresh, non-hemolyzed samples are used for testing. And so this is a phlebotomy consideration that is really adhered to. So let's say you are the phlebotomist and you're having a tough time getting the sample on a patient. You know, maybe you're using a butterfly and, you know, the sample gets, you know, it gets to the blood bank. They're going to centrifuge it. If they centrifuge it down and they see that the serum or plasma is pink to red to start from hemolysis, they will reject that sample. Okay? We need to have normal, nice, like straw colored serum or plasma because, you know, when there's a change and it changes to pink to red when we're doing our testing, that means, you know, hemolysis is a positive reaction. So we can't start out with hemolyzed samples. So the anticoagulants that prevent complement activation in vitro do so by binding calcium. Okay, and so that's our EDTA in this case, in our EDTA tubes. Okay, so when we're talking about routine testing, we have a bunch of different reagents that we use to detect antigen and antibody reactions. And the reactions in vitro, remember, are going to appear as agglutination or hemolysis. And so there are a couple of different methods. Tube testing is the classic way to do the testing. Gel technology is a newer technology that we'll be discussing more later. And there is another one also called solid phase adherence technology. And so tube testing is usually what we start off with in lab, just so you get the idea of antigen and antibody interactions. But all clinical labs have analyzers that use these alternate technologies these days. And we have the gel in our student lab, and I'll show it to you guys hopefully one day. All routine tests use a source of antigen and antibody to detect agglutination or homolysis. So hopefully it's sinking in. The red cell is the source of the antigen if it's there. The serum or plasma, or sometimes a known anti-sera reagent, would be the source of the antibody. And when we combine the two together, if this antibody finds its target on these red cells, that's when we're going to see this sensitization and cross-linking of the lattice formation, and we're going to see the agglutination. If this was not the target, they would just be coexisting in the same tube together. They would not be linked up. That would be negative. So let's talk about some sources of each of these. So sources of antigen, we know it's going to be some type of red blood cell. There are reagent red blood cells that we purchase from manufacturers, and they have a known source of antigen on them. And so these are reagents that we can use to help determine, for instance, somebody's ABO type. Okay. We also have patient or donor red cells, and these are an unknown source of antigen. So, we're using other reagents, we're using sources of antibody to try to find out what antigens the patient or the donor has on their red cells. So we use a commercial anti-sera that's known to help us determine, okay? So our sources of antibody, the reagent anti-sera are known antibodies. They call them anti-sera. It's the same as an antibody. They're made by a company, and they help us determine if somebody has that antigen on their red cells. Once again, when we're using patient or donor serum or plasma, this is usually an unknown source of antibody, and so we might be using our reagent red cells to help find out if the patient or the donor has those antibodies. So this is another picture from our book, and in this diagram, we have a reagent, an antibody against the B antigen that we've added with our patient's red cells, okay? And what we see here at the end, okay, we see the reagent has bound to the antigen. So the question is, what do you guys think? Does this patient have B antigen? The answer is yes. And so we're going to see visible agglutination because the antibody against B found B antigen on the red cells, and we're seeing agglutination in our tubes, okay? If they did not have the B antigen, this would just be kind of out here, not bound to the red cells. Okay, so that's an example of how we use our known anti-sera. So as we go through the semester, we're going to be talking about and virtually, looking at ways of doing a variety of different types of tests. And so this is table 2.7 in our textbook, which basically is just talking about some of the more common procedures that are done in the blood bank lab. And so forward typing is determining the antigens present on the patient's red cells. And so we're going to see if they have A, B, or big D. And how do we find out if they have any of these antigens? We use known antibodies called anti-sera. Anti-A, anti-B, and anti-D. So for instance, if someone has A antigen on their red cells, the anti-A will agglutinate it. If they don't have B, then the anti-B would not be agglutinated. If they had the D, then the anti-D would agglutinate their cells. And so this is how we use these antibody sources to determine which antigens the patient has. The other part of ABO typing is a double check. It's called reverse grouping. This is where we're using known cells from a company, reagent A1 cells, reagent B cells, to determine which antibodies, in this case ABO antibodies, the patient may have in their serum or plasma. Okay, it's the same idea, except what's known is the antigen in this case, and it's helping us find out the antibodies. Antibody screen, we want to determine if the patient has unexpected antibodies, ALO antibodies, that they may have formed from a previous transfusion or pregnancy, and we use reagent cells called screening cells. If we find out with the screen that a patient has some antibodies, and these are not ABO, these are other antibodies, then we move on to identify them, and we do something called a panel. So we use more reagent cells called panel cells. It's kind of fun if you like doing puzzles to figure out which antibodies the patient has. Then the last one here, the cross match, is done with the donor red cells we're thinking of transfusing into the patient. So we take some of the donor red cells and we combine them with our patient's serum or plasma, and in this case we don't want to see agglutination, and that means they're compatible, meaning that if we transfuse these red cells into this patient, we expect a good outcome. And we'll be building on all of these tests as we go through. Okay. Okay, so we're going to finish up this unit and talk about complement. So referred to complement quite a few times previously in this PowerPoint, and I have to make my joke, even though I can't see you guys rolling your eyes at me, but it doesn't mean, oh no, you look great today. It's not that kind of compliment. So basically, complement refers to a system of proteins that are normally present in plasma, and in plasma they're inactive, okay? Or they can be bound to the membrane of cells, and when they're bound to cell membranes, they're active, okay? And so the cell membrane, it could be a red blood cell, but it could also be a bacterial cell, it could be any kind of cell, okay? So I kind of liken complement to coagulation. So if you recall, in hematology, maybe you'd like to forget the coagulation cascade. That was also a series of proteins that, when activated, can obviously the result is a fibrin clot in the coagulation system. This is similar in that one piece feeds into the next piece. They're designated, not with Roman numerals, thankfully, C1 through C9. Similar to many of the proteins for coagulation, they are also synthesized in the liver and also by monocytes and macrophages. Okay, so complement proteins enhance the immunologic process when they're activated. And so what does complement do? It clears immune complexes, those antigen-antibody interactions. This could be taking place on a red cell, or it could be taking place on an infectious agent like a bacterial cell, as I previously mentioned. It is a defense against infection. And what ends up happening at the end is destruction of the cell, cell lysis. So there are two main pathways to complement. The one that is really related to blood bank is the classical pathway. The classical pathway is activated when we have an antibody that is bound to an antigen, so IgG or IgM. And what ends up happening here, remember, the antigen's on a red cell surface, it results in red cell destruction. The alternate pathway, or the alternative pathway, is activated by foreign cell surfaces like bacteria, viruses, fungi, or some parasites. And so basically what happens in this case is destruction of that infectious agent. So regardless of how complement is activated, the end result is the same. It's something called the membrane attack complex, or MAC. which lyses the cell. Okay, and I always joke about this and I say the Big Mac attack, it's going to kill you. Well, yeah, this membrane attack, it's going to kill these cells, whether it's a red cell in the classical pathway or an infectious agent in the alternative pathway. Here we see a diagram of complement from chapter 2 in our textbook, and it's comparing and contrasting classical versus alternative. And so here in the classical pathway, we see, you know, antibody is bound. I wish it looked more like a red cell. It doesn't. Alternative pathway would be an infectious agent. And then there are a variety of different steps that we go through. But the end result here at the bottom, membrane attack complex, is lysis of whatever cell. Okay, and so I'm going to go through in more detail in a chart that I made and talk about the steps in between. Okay, I know it looks crazy and you don't have to memorize this, but I put this together, so I just want to explain it to you guys. I haven't found a good diagram that I like, so I made my own. So we're going to start with the classical pathway, okay? And I'll tell you at the end what's important to know. Classic pathway, remember, okay, this is the one that's important to blood banking. So in phase 1, we have recognition. So we have C1 binds to the immune complex. Okay, remember the immune complex involves the red cell. Okay, and so remember earlier today how we talked about the FC region of an antibody, fragment crystallizable, fragment complement. Okay, that's what C1 is going to bind to. Okay, C1 then cleaves complement protein 4 or C4. And when you see that word cleave, think about a meat cleaver. Okay, it chops something up, right? So whenever you see that word cleave, it's going to break something into pieces. So C1 breaks C4 into 2 different pieces. We end up with C4A and C4B. Okay, and then C4B comes together with C2A, and we end up with something called C4B2A. Do you have to memorize this? No. Okay. But here's what's important. These 2 pieces, the C4B and the 2A, when they come together, they form something called C3 convertase. And you know from your chemistry classes, when something ends in ASE, that's an enzyme. Okay. And so I have a very basic picture that I tried to draw here where you see C3 convertase on the surface of the cell, the red cell in this case. All right? Now, here's something I do want you guys to remember. Any convertase enzyme, no matter what complement system you're looking at, no matter which pathway it is, classic or alternative, the job of a convertase enzyme is to cleave whatever's in its name. So C3 convertase is going to cleave C3 into pieces. Okay? So it comes along, it cleaves C3 into 2 pieces, C3A and C3B. And the piece that we need is the C3B. Okay? So guess what it does then? It takes that C3B and it adds it to what we already have on the cell surface from here. So we already have the C4B, 2A, and now we're adding the 3B. And this forms a new enzyme called C5 convertase. Okay? So what do you think C5 convertase is going to do? Remember, convertase is cleave. What's in their name? It's going to cleave C5 into pieces. So C5 convertase cleaves C5 into C5A and C5B. The piece we need is the B. And so our ultimate thing here, our last phase, phase 3 membrane attack, C5B comes together with C6, 7, 8, and 9. And together, all 5 of these form the membrane attack complex, the MAC, the Big Mac attack. And this is what lyses the red cell. Okay? So what's important to take away from here? Convertase cleaves whatever's in its name. So C3 convertase breaks C3 into pieces. C5 convertase breaks C5 into pieces. Basically, you know, we add a couple things along the way, but at the very end, we have C5b, 6, 7, 8, and 9 come together to make MAC, and that's what lyses the cell. Okay? And that's the classic pathway that is important to blood bank. So just a couple more words about C3b, and I put this on a separate slide because it is important. C3b, you can look at that last slide and say, well, C3b has a pretty important job. It's involved in a bunch of the steps, right? And it's very important. So you guys need to know the functions of C3b. So what does it do? It binds to C3 convertase to make C5 convertase. Okay, we looked at that. It also acts as an opsonin, and you may recall what opsonin means from prior biology courses, but an opsonin is something that coats the surface to attract phagocytic cells. Okay. The last important thing about C3b that's coming up on our next slide is C3b activates the alternate pathway of complement. So we could see that C3b has many important roles, okay? Some in the classic pathway and some we're going to see in the alternate pathway. So let's take a look at that next. Now this looks nuts, I know. So here we have our alternate pathway. Remember, it does not require antibody for activation. It's a natural defense against infection, okay? So we have C3b that spontaneously forms and binds to some foreign cell, bacteria, virus, what have you, okay? We have something called factor B that binds to that C3b. And together, they make factor D, and factor D cleaves factor B into pieces, and you can see there's an A and a B piece. Please don't worry too much about these details, but what's important to note is that all of this breaking apart and things coming together, we end up with something called C3b, big B, little b, and this is known as the C3 convertase of the alternate pathway. So it's composed of different pieces of complement, but it's important to note that alternate pathway and classical pathway do have their own variety of C3 convertase. And so what does C3 convertase do? It's going to cleave C3. We're going to see that next in step 4. So C3 convertase cleaves, breaks more C3 into A and B components, okay? And then it's going to take the 3B and add it to this original thing, okay? Similar to what is done in the classic pathway, okay? Components are different. We have C3b, big B, little b, 3b. That is the C5 convertase of the alternate pathway, okay? What is C5 convertase going to do? It's going to cleave C5. It may be composed of different pieces, but it has the same job. It's going to break C5 into A and B. And then once again, just like the classic pathway, just like coagulation where it ends up in the common pathway, they end up in the same thing. C5b with 6, 7, 8, and 9 come together to make the membrane attack complex. This coats the cell surface. In this case, it's a bacterial, viral cell, what have you, and causes it to be lysed. Okay? So let's compare and contrast. Both arms of complement, classical and alternate, both have a C3 convertase that cleaves C3 and a C5 convertase that cleaves C5, even though they may be composed of different pieces, and they both end up in C5, 6, 7, 8, and 9 forming MAC, and MAC basically destroys the cell. Okay? Classical is used mostly with antibody, immune complex initiation. Alternate is done with infectious agents. That's basically what you need to know. All right, and so here we see, some of the effects, table 2.4 of complement. And so we talked about opsonization, which enhances phagocytosis. our neutrophils responding, etc. Okay, kills, you know, we talked basically about all of this. Okay. So just a little bit more. I think this may be the last slide. Thankfully, just like we have natural anticoagulants to control the coagulation cascade, there are also normal control mechanisms to prevent overactivation of complement, okay? And most of these regulate C3, and we saw how C3 is really important in both pathways. So regulating C3 is a good way to place some controls on the process. Complement can have harmful effects and result in damage to host cells and tissue in certain instances, okay? So remember, its job is really to destroy foreign red cells or to destroy infectious agents, but sometimes there can be a deleterious effect to the person. So when someone has an overwhelming gram-negative septicemia, maybe you recall from phlebotomy, this is a bloodstream infection that can be life-threatening. And so when complement is overactivated, sometimes host tissue can die in the process as well. When it's activated by tissue necrosis, necrotic means dying tissue, can have deleterious effects. Or when there's an excessive amount of immune complexes. This is a typo, the 67 shouldn't be there. I'll tell you guys a secret, my cat walked across the keyboard, I'll get rid of that. But when there's an excessive amount of immune complexes, complement can be overactive and there can be damage to the host as well, okay? Sometimes people can have complement deficiencies and these can be inherited, and when they do, that person has serious excessive infections that can be life-threatening. So think about it. That alternate pathway, that's how really we get rid of these infectious agents. If someone has deficiencies, they're at risk of dying from infection. And lastly here, complement can be measured in the lab, and usually they're measuring levels of C3 and C4 components to make sure somebody has an adequate amount. Okay, so I know this was a lot of information. Please listen to this as many times as you can stand. We have to start somewhere, and so this is the beginning. We'll be building on it. And please remember that after each PowerPoint unit, there will be a quiz on the information that you've listened to. So when you've listened to it enough, please make sure you take the quiz. Thank you.